Today, I would like to talk about bovine virus, diarrhea virus infection disrupt maternal pregnancy recognition in cows. And firstly, is the background. Poor fertility causes economic losses to the dairy industry. Early embryonic mortality is a major component and is at least in part caused by the failure of the pregnancy recognition. In the cows, pregnancy recognition is initiated by interferon tau released from the concept acting on the uterine endometrium. This would lead to the inhibition of luteolysis by preventing the parasital release of prostaglandin F2 alpha and switch the PG production to PGE2 and stimulate expression of type 1 interferon stimulated genes. This creates a receptive environment for the implantation. BVDV causes widespread infection in cattle worldwide. The reproductive system, including the uterine endometrium, is a major site of infection, maintenance, and the spread of the virus. BVDV infection may have a significant impact in all stages of pregnancy, including causing early embryonic death, such that reproductive losses are one of the most important consequences of the disease. Our previous evidence suggests that the BVDV infection may cause the PR failure. Our hypothesis is that the BVDV infection disrupts pregnancy recognition by altering PG and ISG signaling pathways in the uterus. And this study is to investigate the effect of BVDV infection on the production of important maternal PR components, including uterine prostaglandins and the interferon stimulated genes, using an in vitro PR model. We collected the uterus from 10 cyclic cows and cultured it with the DMEM F12 medium, including BVDV-free fetal count serum. And day 4, and this cell culture will be about 70 to 80% of confluence, and we infected the cells with the NCP-BVDV with the PE515NC strain. And there are 2 plates. One is for control, and one is for the BVDV, because if we use the same plates, it will be cross contamination. And day 8, we treated the wells with 100 nanogram of interferon tau, and all this will generate 4 combinations of treatment. First is the control, and the IFNT alone, and the combination of BVDV with the IFNT and the BVDV alone. On day 9, we harvest the cells for the extraction of mRNA and for the analysis of the proteins. And firstly, we confirmed the infection was successful. This is the DNA ladder. This is a BVDV positive. This is a vector of BVDV virus positive. And these 2 lanes, lane 3 and lane 4, are the treatment without BVDV, such as IFNT alone and the control. Lane 5 and 6 is the treatment contain BVDV and the BVDV plus interferon tau. This confirms the infection of the BVDV to the cells were successful. Firstly, we analyzed the effect of treatment on prostaglandin production. In this side are the prostaglandins. This side are the enzymes for the synthesis of the prostaglandins. We can find that here the BVDV the interferon tau stimulation increased the production of PGE2, although the BVDV increased the PGE2 production as well. But in the cells infected with BVDV, the stimulated effect of interferon tau lost, and the effect of interferon tau was inhibited by the BVDV infection here. We can say here is the PGF2 alpha. This is the BVDV have a slightly effect in the production of the PGF2 alpha. And this led to the increase of the ratios of the PGE2 to PGF2 alpha by the BVDV virus. And this side are the enzymes we correspondingly, and for the cyclol oxygenase 1, interferon tau and the BVDV increased the expression of PTGS1, namely COX1. And in the cells infected with BVDV, the stimulated effect of interferon tau lost and neutralized. So no differences between the control and the interferon tau plus MCP BVDV. And for the COX2, the effect of treatment are very slight. And for the PGES1, we can say here MCP BVDV alone significantly stimulated the production of PTGES1. And this is the AKRKB1, is the synthesis of PGF2 alpha. We can say the effect of treatment are moderate and slight. And only the combination of interferon tau and the BVDV infection inhibited the production of PGF2 alpha synthesis. This is the effect of the treatment in the receptor of prostaglandins. This is the prostaglandin E2 receptor. This is the E3 receptor. And we can find the interferon tau significantly stimulated the expression of prostaglandin E3 receptor. But in the infected cells, you can say the stimulated effect was significantly inhibited. And this is the effect of the treatment in the upstream prostaglandin production pathway, including PGR, progesterone receptor, estradiol receptor, and the oxytocin receptor. We can find the treatment on this receptor are moderate, and all of them insignificant effect of the insignificant. And next, we tested the effect of treatment and the ISG interferon stimulated gene production. The first four are the genes involved in isolation. Isolation is important for the immunity and pregnancy recognition. The ISG15 and HERC5. We can say for these two genes, interferon tau significantly stimulated the expression of ISG15 and HERC3. But in the infected cells, BVDV infected cells, this effect are inhibited, significantly inhibited. And for these two genes, they have a different pattern. And we can find the interferon tau in the BVDV in the infected cells, both AFNT and the combination increased. the production of USP18. And this, for this one, is the old treatment increased the production of TRIM56. This is another 6 genes, including DDX58 and IFIH1 and IFIT1, IFIT3, and BST2. We can find all the 5 genes following the same pattern as we talked previously as the ISG15. The IFNT increased the production of those genes, but in the infected cells, in the BVDV infected cells, the effect was inhibited or neutralized. And this is another 4 interference stimulated genes, including MX1, MX2, and RSAD2, and OAS1Y. We can find it following the similar pattern. This is interferon tau increased the production of those genes here as well, but in the infected cells. And the expression of this gene, the stimulated effect, the effect of IFNT was inhibited. And the next are another 4 ISGS, SAMD9 and the GBP4. We can find for these 2 genes following the similar pattern, the interferon tau, the very, very big effect, and this in the stimulatory effect, and in the BVDV infected cells, and the effect inhibited. And for the IL1B is not ISG, but we use it for to monitor the general immunity of the cells. We can find the interferon tau inhibit the IL1B production. This is important for the pregnancy recognition because it has to keep the uterus quiet to prepare for the pregnancy. But for the combination of the IFNT and the BVDV infection causes the big disrupt for the expression of the IL1B. And next, we tested the regulatory pathway in bovine endometrium for the ISG production. Finally, this is the pathway for the ISG production is IFM alpha act as a receptor in the effect of TYK2 and JAK1 and the phosphorylation and combined with the state 1 and state 2 and then become and combine with the IRF9 and become a complex and enter the nucleus to cause the production of ISGS. We pick up the key genes in this pathway to test. And for the receptor 1 and the IFN receptor 2 and the JAK1, we can find the effect of treatment very slight, moderate. Only IFNT have the significant effect and the receptor and the IFN alpha receptor 2. But for the TYK1, we can find the both BVDV and IFNT stimulated the production. But it's interesting to say in the BVDV infected cells, the effect of IFNT was inhibited. Here as well, the effect of the IFNT inhibited for the genes of IRF7 is interferon regulatory effect 7. For the regulatory effect 9, the family following the similar pattern, we can say the IFNT stimulated the production, but in the infected cells, this stimulatory effect was inhibited. And also, BVDV infection alone also decreased the expression of the IRF9 This is state 1, and state 2 is an important pathway. And if you find the similar pattern, IFNT stimulated the production of state 1, and here as well state 2 as well. But in the infected cells, this effect was neutralized, inhibited. And this is the interferon gamma. It is not an ISG, but we use it to monitor the general antivirus effect of the cells. We can find that during the IFND treatment and the BVDV infection, the expression of interferon gamma significantly increased. And in order to confirm the translation of the gene expression to the protein, we measured the ISG15 in the culture medium and the state 2 protein in the cells. We can find it following the similar pattern as the most of ISG expression during the treatment. IFNT treatment significantly increased the production of ISG15, but in the cells infected with the BVDV, the stimulatory effect was significantly inhibited, the similar pattern of the state 2 protein as well. And the next is the summary of our experiment. This is during the pregnancy recognition, the concept released IFNT and act on the maternal IFN alpha receptor and led to the activation of signaling pathway. It's the state 1 and 2 signaling pathway, and to cause the production of ISG. But during the BVDV infection, all this part was interrupted. It stopped the production of the activation of the state 1 and 2 pathway, and they also inhibit the production of the ISGS. This led to the decrease of the number of the pregnancy specific ISGS. So this led to the decrease of the uterine immunity and the interrupt of the implantation, and also this is it disrupted the pregnancy recognition. And in this side is the prostaglandin pathway. During the pregnancy recognition, and the inhibitor of the BVDV infection inhibit IFNT stimulated IGF, stimulated the PGE2 production. This would directly lead to the implantation and the PR failure. Before the pregnancy recognition, BVDV increased the basal ratio of PGE2 to PGF2 alpha. This would decrease the uterine immunity and led to the various uterine disease. This would in turn lead to the pregnancy recognition failure. And finally, I wanted to thank all my colleagues involved in this project. All of them have a very significant contribution to this study. Thanks.